Okay, well, thank you very much for having me for this wonderful workshop. It's day 4. Just to tell you where I come from, I work at the National Cancer Institute, and like many of the faculty here, I run a core laboratory or a shared resource laboratory in the NCI in Bethesda, Maryland. We are not a diagnostic lab, but we support both basic and clinical research. We do a lot of clinical trial work where I do. And as we have mentioned earlier, building and modifying machines is a big part of what we do in my laboratory to try to apply to special purposes. Before I get started on proliferation, and I won't talk for a real long time because we want to stay on schedule, but I just want to remind everybody that this is not the first Montevideo flow workshop. We had one in the spring of 2022, a remote workshop. And we had great, we had Roxana was there. Marielle, of course, was running the workshop. This website is one of many websites we created for all of the remote workshops we did. It is still online, and this was just one of many that we did. This was a wonderful 2-day spectral workshop that Maria actually and Rui Gardner at Memorial Sloan Kettering ran. All the content is still out there as well. This is an Indonesian clinical cytometry workshop that was organized by Paul Hutchinson in Singapore. And I'm bringing this up because during the pandemic, we had a lot of remote programs and it was all recorded and everything is still online. Sasha talked about this early in the meeting, that the fact that this content is all still out there and we don't want it to get lost. I mean, we're all trying to forget the pandemic now, but so much of the material is out there by some wonderful speakers. So what we have done is created a couple websites. We have one organized by conference, and there's the Montevideo workshop down there, but there were probably about 15 or 20 workshops. All the slides are there, all the video is there. And we're starting to curate it. So we're breaking the talks up into individual meetings or individual subjects on basic flow, multicolor flow. The make your own is there. The spectral stuff is there. So we'll give you the URLs for these. But if you are interested, go and take a look at it. We want to make sure that this content doesn't get lost. Because during the pandemic, we could invite anybody from anywhere in the world. We had some of the best flow cytometry people anywhere. And all this material is still out there. It's absolutely free. We will keep it up forever, and we want to make sure it's archived and preserved. So do feel free to go in and explore it. All the slides are there too, not just the video, but the slides. Also, for those of you still interested in the flow cytometry, the build your own flow cytometry lab, we have a website for that as well. And we also have an hour-long video that we hurriedly prepared at the beginning of the pandemic for Cyto. We need to re-record it because it's a little bit rough, but that is still available and is still free as well. So if you visit our website, you can actually download all the video sections of that. So for the last 3 days, we've talked mainly about phenotyping and high dimensional analysis and spectral analysis. I think the talks today are going to cover other applications for flow cytometry. I'm going to talk about proliferation. This is something I have done for many, many years, but I'm really not an absolute expert on it. So the talk I'm going to give is composed of work from a number of people. And in fact, it's based on the talk designed by Kylie Price for the Cyto lecture series that is available on the ISAC website. Some of you here know Kylie. Kylie runs many laboratories now, not just the flow cytometry laboratory, at the Mulligan Institute in New Zealand. And I'll also be presenting data. Lola has some data in this presentation. Paul Wallace at Roswell Park, who has developed a lot of techniques for proliferation, also has data in here. So what you're seeing is data from a number of places. This is Kylie at the Mulligan, and Kylie got to meet the Prime Minister of New Zealand a couple of years ago, which was a nice, her institute is important enough that the Prime Minister decided to visit. So Kylie is a very good friend to all of us and is a big force in the flow cytometry world. This is Paul Wallace, who's also a good friend to many of us. This is at the most recent Nepal workshop in a selfie with the students. So they have both contributed a great deal to this technology, and much of the data you're going to be seeing today is from them. So that remember, when we're measuring proliferation, we're looking basically at cell cycle progression that ultimately leads to cell division. And in the immune system, this is usually in response to some sort of stimulus. In tumor cells, we're often measuring dysregulation of cell cycle. So the things that we want to measure are cell cycle progression, going from the classic mitosis cycle from G0, G1 to S to G2 and M phase. We're looking at things like doubling time, proliferation rate. We're looking at the stochastic nature of proliferation. Remember that if you stimulate 100 cells at the same time, not all of them will immediately proliferate. Some won't proliferate at all. Some will proliferate slowly, some will proliferate rapidly. And we now have techniques to look at this heterogeneity, the so-called stochastic nature of activation. Also, we can look at disruptions in proliferation, which is important for things like cancer cell research. And we have a lot of techniques that will allow us to do this. So these are the techniques I will cover in the next 45 minutes. We can do cell cycle, which is one of the oldest techniques in flow cytometry, but still very useful. We can immunolabel for proteins that are associated with cell cycle. We can do the same multicolor analysis we do for surface phenotyping. We can also look at intracellular cell cycle proteins as well, and we can combine it with DNA cell cycle to get a multi-parametric, multi-dimensional picture of cell cycle. Many of you are probably familiar with nucleotide incorporation assays, BRDU and EDU labeling assays. We'll talk about them. Dilution proliferation assays. And there are also techniques using fluorescent cell cycle specific fusion proteins to look at cell cycle as well. Zbigniew Darzynkewicz, who very sadly passed away 2 years ago, is a real pioneer in this area. He's a good friend of myself. He was a good friend of myself, to Zasha, to most of us in the flow cytometry world. He was one of probably the 10 people that founded flow cytometry. And a lot of the techniques we use for DNA cell cycle proliferation were developed by or improved on by him. So you'll be seeing his work throughout this talk. So many of you are already familiar with cell cycle analysis. It's really one of the oldest techniques in flow. You take a cell, you permeabilize it, and you add a relatively high concentration of a DNA binding dye. It binds stoichiometrically to the DNA. And then if we analyze the fluorescence distribution of the dye on a linear scale, we can visualize the G0, G1, S, and G2M phases of the cell cycle. We can permeabilize the cells with ethanol, with detergents. We can also extract nuclei for very high resolution analysis, so the DNA dyes don't bind non-specifically. But then we lose the cytoplasm. And one of the nice things about cell cycle analysis is that you can combine it with immunolabeling for other proteins to get a richer, deeper understanding of what is going on during the process. So you can see here very clearly, we have cells in G0, G1. As the DNA replicates, they enter S phase and the content increases. We get to G2M and ultimately the cells go back to G0, G1. This is probably one of the first techniques that we ever did in flow cytometry 40 or 50 years ago. Still very useful today. There are different ways to permeabilize cells. We can extract the nuclei using hypotonic buffers. Citrate buffer is very common for this. We can use ethanol treatment. I'm saying fixation here, but it is not really fixation. Ethanol does not fix cells the way formaldehyde or glutaraldehyde does. But it will poke holes in the cell membrane while still preserving the structure of the cell. However, intracellular proteins and fluorescent proteins being expressed in cells are often lost during this process. So we can also use fixation and detergent treatment to permeabilize the cell. This tends to degrade the cell cycle resolution a little bit. The fixatives cross-link DNA and inhibit binding of the DNA dye. But sometimes this is a necessary price to pay to get intracellular protein expression. We can do this with cell impermeant DNA dyes, but we can also do it with cell permeable DNA dyes like Herx 33342, DRAC5. The cell cycle is lower, but this gives you the potential to then sort the cells and put them back into culture. The cells don't have to be killed to do your cell cycle analysis. Now, you can use almost any DNA dye. Propidium iodide is historically the most common. It's the one we'll be using today. PI binds, however, not only DNA, but also double-stranded RNA. So you need to treat your cells with RNAs to make sure that you remove that factor and are only seeing DNA content. The big problem with using propidium iodide, however, is that it is spectrally difficult to combine with other dyes. It has a very broad emission spectra. So if you are going to be doing additional labeling, you often need to consider other dyes. It's just like selecting probes for doing multicolor flow that we've been doing for the last 3 days. You have to select the dye with the best spectral properties to be compatible with the other dyes you are using. 7-AAD is also blue excited, like laser excited, like PI, but it has a longer red emission and is often used for cell cycle. If you have a UV or a violet laser, and you do have violet lasers on your machines, there are several dyes out there that can be used. DAPI is probably the most common. It does not bind RNA, which makes it very useful for cell cycle analysis. There are red excited dyes, and in all cases, there are cell permeable dyes out there that do not require fixation or permeabilization. But you can really use almost any DNA dye. So this is the sort of thing we can detect. These are HL60 cells showing very classic, these are ethanol treated and labeled with propidium iodide, showing a classic G1 SG2 distribution. However, in this case, I've irradiated the cells with gamma radiation. And when I do, you can see the cells become arrested in somewhere between the beginning and the end of the G2 phase of the cell cycle. They replicate their DNA, but they don't return to G1. They don't ultimately divide. And we can detect this very readily by looking at the propidium iodide DNA content. We have very few cells in G0, G1. Everything is accumulated up here in SNG2. A classic cell cycle experiment to look at cell cycle perturbation. However, this is only one parameter, and we don't know exactly where the block is. It could be at the end of S phase. It could be in G2 phase. It could be immediately before mitosis. So this gives us information, but it doesn't give us a lot of information. Now, remember that DNA dye labeling needs to be stoichiometric. If you have used DNA dyes as viability markers, you use a much lower concentration of the dye than you do in cell cycle. For viability, all you have to do is just get a little bit of DNA dye in there to see where the dead cells are. We have to use a lot more. In our case, we use 50 micrograms per mil of propidium iodide. We need to saturate the DNA dye. We don't want to label too many cells. Probably the most common source of poor cell cycle labeling is having too many cells or not enough DNA dye. We have to saturate the chromatin. We'll be excluding doublets using something called doublet discrimination. You've already seen this, I think, in a lot of the analysis we've been running so far in scatter. We're going to use doublet discrimination of DNA fluorescence to make sure that we remove doublets or triplets. These can mimic a 2G1 cell stuck together can look like a G2 cell and disrupt your data. We want to make sure we're not including those cells. We'll also be using specialized software to model the DNA content. I've been showing you brackets here. This is really not correct. We understand when we run our DNA distributions that in fact our S and our G0, G1 phase cells overlap with one another in a single parameter, and the same for S and G2M. We need to use curve fitting software to get an accurate percentage for the G0, G1, S, and G2 phase cells. Fortunately, most of our flow cytometry programs can do this now, FlowJo, FCS Express. There are also more specialized programs like ModFit that are more powerful, that have many of these curve fitting models and are very good for more complex cell cycle problems. They're expensive, unfortunately, and ModFit only does one thing, which is cell cycle. However, you may need to use it for more complex analysis problems. So this is what some data looks like. We ran this, Karen and I ran some cells yesterday on one of the machines we're going to be using today for DNA content analysis, the ANII. So this is fresh data, only 24 hours old. These are EL4 cells. EL4 is a mouse lymphoma cell line. It's chemically transformed, not viral, so these cells are quite safe. EL4 is like HeLa cells. It's an ancient cell line. So we determined the scatter. You can gate on the scatter, but you don't always want to necessarily, because there may be proliferating cells up here outside our normal gate. Remember that this is a cell line here. So these were cells that were treated with ethanol, labeled with propydium iodide. We then did what we call doublet discrimination, where we look at the DNA content, but we also look at another parameter that will tell us whether there is more than one cell present. On the ANII, we have to use pulse height. Both, I think Roxana talked a lot about area versus width versus height in her presentation. Many machines also provide width measurement, which is actually better. But not all systems provide that. So we exclude the doublets. The doublets would populate up here above with a higher height measurement, but a similar area measurement. And here's our cell cycle measurement over here, analyzed using a model. I think this is the JET model over here, the curve fitting model, done within FlowJo. So the program estimates G1S and the G2 populations over here. It also calculates things like the ratio between the G1 and the G2 peak. This should ideally be 2. It isn't always 2, but we hope that the cells that are in G2 M phase, pre-M phase, are going to have double the DNA content that the G1 cells have. Also, remember, we are now on a linear scale. We don't use linear scales very much for fluorescence and flow cytometry, but cell cycle is one application where we do, where we need to. Now, DNA content can be much more complex if you're looking at a cancer cell population. Aneuploidy, when a cell has an abnormal diploid content, is often observed in tumor cells, sometimes in the real world too. And you can end up with multiple cell cycle populations where you have the diploid cells here, the aneuploid cells here, and then you have both diploid and aneuploid S and G2 populations over here. This gets complicated, and sometimes you need more sophisticated software to analyze it. We're also starting to use, in this case, internal controls for DNA content. Like any other technique in flow cytometry, it's good to have controls that tell you where your diploid population is. And in a case like this, in a tumor line, we may not necessarily know whether the aneuploid cells here are in fact diploid or not. We have two major populations that are both G1. What we can do is take biological controls that have nuclei. Chicken and trout erythrocytes are common for this. Chicken erythrocytes, for example, have 30% of the DNA content that normal human cells do. So we insert some chicken red blood cells into our sample, and we're able to determine precisely where the diploid population is, and then anything above that is non-diploid. Cell cycle can get complicated. We're not going to talk about plants very much in this workshop, but plants do weird things in their cell cycle. They can become triploid, they can become tetraploid, polyploid. Plants have evolved many strange strategies for enhancing their productivity, And many of these conditions are economically important. Plant growers do a lot of cell cycle analysis. They want to, it turns out that tetraploid, triploid, and tetraploid plants are more productive than diploid. You also may need more sophisticated DNA modeling software for a situation like this that will go in and identify the diploid and the aneuploid population. Now, that's one parameter cell cycle. However, we can also label our cells simultaneously for a variety of proteins that are important checkpoints in cell cycle. For example, these are cells that are labeled not only for DNA content, but also for a cyclin B1. Excuse me. If you know cell cycle, you know that cyclin B1 is expressed in mitosis and then is immediately degraded once the cells return to G0, G1. So in this case, I have permeabilized my cells with a detergent, and then I've labeled with an antibody against B1. And instead of one parameter plots, I now have two parameter plots where I am looking at G0, G1, S, and G2, but also cyclin B1 expression. And you can see in the untreated cells, we have, as expected, cyclin B up. But then it immediately degrades once the cells return to the G0, G1 phase. If I irradiate the cells and the cells become arrested, the cyclin B1 expression goes up very strongly. And we now know the block isn't back here at the beginning of mitosis or in G2, but is in fact very late in mitosis, immediately prior to cell division. So by doing multi-parameter flow, just like for phenotyping, you're getting more information by putting additional markers in. And this can actually get quite sophisticated. We can look at a variety of markers. This is actually data from Dr. Darzenkevich's lab and also from Jake Jacoberger, who's at Case Western Reserve, where they are analyzing for cell cycle on the x-axis, but then a variety of cyclins. Cyclins are expressed and degraded at different points in the cell cycle. They're important regulators of cell cycle progression. Cyclin D1 comes up in G1. Cyclin E is a late G1, early S phase marker. Cyclin A comes up in S phase, and then B1 in G2 and G2M. So we're getting a lot more information here than we would from just DNA content alone. There are hundreds of proteins we can look at: cyclin-dependent kinases, cyclin-dependent kinase inhibitors, many, many things. And we can combine multiple checkpoint markers into a single sample, just like we can do multicolor flow cytometry. So we can get a lot of information about what's going on. This has even moved into the clinical world. Ki67 is a DNA polymerase dependent ribosomal RNA synthesis protein. It is often used in clinical flow to assess proliferation. Proliferating cell nuclear antigen is expressed throughout the cell cycle, but in rapidly cycling cells, it comes up in S phase, and it's also looked at as well. Remember, these markers are intracellular. You have to poke holes in the cell to get them in, but you need to do it in a way that will also preserve the cell cycle. The protocols for all these are very specific and must be closely followed. But there's a lot of this in the literature, and either you can find the technique to label your cells for the target protein you're interested in, or you can use those protocols for guidance in designing your own protocol. Some of these targets are cytoplasmic, some are nuclear. Different techniques are used for labeling cytoplasmic and nuclear proteins. And often we combine cell surface immunolabeling as well, so we can be putting in surface markers along with our intracellular markers. We can look at other proteins as well. This is also from Dr. Darzenkevich's lab. He was very interested in looking at DNA damage-associated proteins: phosphorylated histone H3, gamma H2AX, ATM. These are all associated with DNA damage. Cells express these when they're exposed to ultraviolet light and other environmental insults. In this case, he's looked at DNA content versus gamma H2AX expression, and you can see how ultraviolet light produces a lot of gamma H2AX expression in S phase. So again, a very powerful technology. H3, phosphorylated histone H3, as you would expect, becomes very dominant during mitosis. So it's expressed very strongly there. So that's DNA content. Moving along, another technique you may be familiar with is thymidine analog incorporation. Although cell cycle does give us a lot of information about what's going on in the cell, it doesn't really provide any kinetic information. Conventional cell cycle analysis is a photograph, an instant in time. By using thymidine analog incorporation, we can find something out about the kinetics of cell cycle. So this is a technique you may have encountered before. What we do is we take our cells, we incubate them with a thymidine analog. Historically, this has been bromodeoxyuridine, an artificial uridine substitute. We can just add it to the cells, or we can pulse the cells with it, add it for 1 hour, and then wash, get rid of it, and allow the material to start entering the cell cycle. We then permeabilize our cells. We treat the DNA with acid or DNA, something to break up the structure a little bit, and then we have an antibody that will go in and bind to the BRDU. We can therefore look at BRDU incorporation into the DNA, S phase cells, the cells that are where the DNA is replicating, and get an idea of how many cells are entering S phase, whether the cells are proliferating or not. So this is what the data looks like. This is our cell cycle up here. Remember that we are combining it with DNA cell cycle, but we are also looking at our BRDU incorporation. So the G1 cells are down here, and the BRDU is being incorporated and detected in the S phase cells. Now, this is a relatively short time point, so we're not yet seeing it in the G2M phase cells. But ultimately, if we continuously incubate with bromodeoxyuridine, all the cells are going to become positive. So this is a kinetic technique. We can start to get information about what rate the cells are entering S phase, the proliferation index of the cell. You can use BRDU in cell culture. It's been actually widely used to look at cell cycle and proliferation in vivo by adding it to the drinking water of laboratory animals. Very common technique that's out there. Now, BRDU has been used for many, many years. However, there are problems with it. You have to disrupt the DNA structure to get the antibody in, and this can cause trouble with DNA content labeling. So there's really a better technique now called EDU Click-It. It's a small molecule technology where you incorporate a similar nucleotide analog and then use a small molecule coupled to a fluorescent probe that can enter the DNA helix without disrupting the DNA. Click-It is I believe it's a Thermo Fisher trademark, and this has largely replaced BRDU. It's a much gentler technique and easier to use. But the result is very much the same. So when you're doing these techniques, and this is true of all of the proliferation techniques we're going to talk about today, a lot of optimization needs to be carried out. You need to titer the amount of bromodeoxyuridine. You want to use the lowest concentration possible. If you're doing BRDU labeling, the DNase treatment, which is what we use to break up the chromatin, also has to be titered. You don't want to damage the DNA so much that you don't get good DNA content measurement. However, if you're able to, I would use EDU Click-It. It's really, really the dominant technique. However, people have combined both BRDU and EDBU in pulse chase experiments to get an even deeper picture of proliferation, since they can be detected independently from one another. However, one problem with this technique, if you're not pulsing, is that ultimately the cells all become positive because your cells are continuing to cycle and they will all become positive. So we can do pulse chase experiments. This is actually one from, I believe, from Kylie, where they're using a human glioblastoma cell line. They're pulsing the cells and then looking at intervals. And you can see the material entering the cell cycle. It enters S phase, then G2M, then it ultimately cycles back to G0, G1. Okay, but because of the fact that ultimately the entire population becomes positive, we're getting more information than DNA content, but we're still not getting the full picture of the random stochastic nature of cell cycle analysis, of cell cycle. So that brings us to our next technique, DNA dye proliferation assays. The principle behind these is very simple, and again, many of you may have already encountered this technique. What you can do is take your cells, label them with a membrane or a protein dye, not a DNA dye in this case. The dye sticks to the outside of the cell or enters the cell. And then as the cells divide, the dye is divided. So after one cell division, the dye is divided in half, then a quarter, then an eighth, and then a 16th, and so on. So we're not seeing only one round of replication. We can see 2 or 3 or 4 or 5 or 6. This is a little cartoon from Alice Gibbon, who used to be at Dartmouth University. She's now retired, but she was a pioneer in this technique. So if you have a population where only one of your cells is proliferating, you can get an idea of that fact, that you will have cells that are not dividing. They remain fluorescently very bright, and then ones that are progressively undergoing 2, 3, 4 rounds of replication. The heterogeneity of proliferation becomes obvious in this technique. So this is what the data ideally would look like if it were a perfect world. We would start out with our initial labeling, and I'll talk in a moment about the types of dyes we use for this. And then after 1 round of replication, 2 rounds, 3, the fluorescence decreases. This is what the data looks like in the real world. These are 2 different types of dyes, a protein dye and a membrane dye. I'll talk about both of them. But you can see the data still looks pretty good. Here's our first generation, second, third, fourth, fifth, sixth, seventh. And we get a very, very nice view of proliferation. Really, if you're publishing a paper these days on cell proliferation, your reviewers are going to want you to use this technique. You can use bromodeoxyuridine in cell cycle and other things, but there's a lot of information contained in this data. This is a protein dye. This is a membrane dye that gives the same phenomena, and we'll talk about both techniques. So 2 types of dyes. There are membrane dyes. The PKH dyes are the most common ones, but there are others now as well. These are dyes that intercalate stably into the plasma membrane. You add them to cells, they insert into the membrane, and they remain there. So when the cells divide, they are divided between the cell, and the DNA, the amount of fluorescence decreases. There are also protein reactive dyes that will stick to sulfo or amine residues on proteins inside or outside the cell. And if you've done these techniques, this is the one you may have heard the most about, CFSE-DA. It's similar chemically to the dyes we use to conjugate our antibodies. They're highly protein reactive. They will stick to the cell stably, and then again, they'll be reduced as the cell populations die. Now, this is not a perfect phenomena. Of course, dye can be lost by passive diffusion. 2 cells sticking together will exchange the dye. But good technique design can minimize these problems, and you can get some very good data out of this. So the membrane dyes are the oldest ones. These have been around for quite a while. The ones that we're most familiar with are the PKH dyes. You can look at their chemical structure over here. They have these long hydrophilic chains that insert into plasma membranes and stay there. And the longer the chain, or the greater the number of chain, the more stable the dye is in the cell. They're available from Millipore Sigma. Thermo Fisher has some. There are some other ones out there. You have to incubate these cells, these dyes in the cells under slightly hypotonic conditions. These dyes don't want to get into the membrane normally. So we do a very, very brief incubation, minutes, in a buffer that is slightly hypotonic that will encourage the dye to incorporate. And then we put the cells back into isotonic conditions. They're relatively non-toxic because they don't covalently bind to anything. They just stick in the plasma membrane. But all of these dyes can be a little bit toxic at high concentrations, or they can inhibit cell growth. Cells don't like to be labeled with these things. All of these techniques have to be worked out. You will not get them right the first time. You have to titer the dyes and the incubation conditions and other things. And some of them are good for short-term incubation. They tend to be gentler to the cell. Others have longer chains and are better for long-term incubation, but they may be a little more disruptive to the cell population. Now, the protein dyes, CFS-EDA is the most common one. These are protein reactive. They cross the plasma membrane, and then they bind non-specifically to proteins, to amine residues on proteins. So different mechanism, but similar idea. You're trying to introduce a stable label into the cells. For the protein dyes, CFS-EDA is the most common. Cell trace, I think, is now the trademark name that Thermo Fisher uses. It's excited at 488 nanometer. It emits in the green range. It is extremely bright. However, there are red and violet excited versions because you may be combining this with other markers, extracellular or intracellular markers. Like all the flow we've been doing for the last 3 days, it's not just about one marker, it's about all the markers. And you may need to do some panel design to incorporate these into, into your analysis. We often combine proliferation markers with extracellular and intracellular labeling. These are extremely bright. However, cells can shed them back out into the plasma membrane, into the extracellular media. It's something we have to think about. Because they covalently bind to proteins, they can also be somewhat toxic. And one of the things you'll need to do is titer these things down to the lowest concentration you can use and still get nice peak distributions. So you'll have to do a lot of testing. You will not get it right the first time. Now, I won't spend a lot of time on technique. I've included the slides because you'll be getting copies of these slides. But there are very detailed methods for all of these dyes. This is from Paul Wallace's and Joe Terrio's group at Roswell Park, where we are labeling membrane dyes. Part of the problem with these dyes is that they label cells immediately, which means you have to mix them very, very rapidly. And there are little tricks for doing this to get uniform labeling. But follow the techniques, follow the protocols very closely. There's a nice Jove video presentation about this, a very nice one out there. This is for the protein dye incorporation. And again, the technique is very fiddly. It's very precise, and you need to follow it very precisely. So this is what the data looks like. We have a protein dye here, a DNA dye over here. We always take a time zero. We want to know where the cells are going to begin. And then we'll look at the generational peaks down here. Now, the membrane dyes tend to be less precise in terms of their peak resolution. You can often, you don't get this nice single peak resolution. Is that a problem? And there have actually been a lot of studies on this. However, we use the same curve modeling and fitting programs for dye incorporation that we use for DNA content. And in fact, this is not an enormous problem. The software can still go in and do a pretty good estimation of what your generation 2, 3, 4, 5 peaks are, so on, even if the peak resolution is not perfect. This is a study, I think this actually, Kylie gave me this data, but I think this came out of Paul Wallace's lab where they tested protein versus membrane dye and essentially got very similar results, even though the peaks are not as well resolved. So you can really use both techniques, and depending on your cell type, the system you're working in, you may want to try both techniques. This is an old experiment from my lab. Back in the 1990s, I worked with a group that put chick chondrocytes on the space shuttle, STS-95, in cartridges to look at cartilage deterioration in zero gravity. So prior to inserting these cells into these cartridges that went up in this module that sat in the mid-deck locker on one of the space shuttles, we labeled the dyes with a PKH dye to look at a gradual decrease as the cells divided. And even by day 17, we still had some fluorescence in there. They're well retained in cells. However, we had to be careful what dye we chose. PKH2 is a short chain dye. It's good for short-term proliferation analysis, but it doesn't stay in cells for very long. We ended up going with the longer chain dyes to do that particular experiment. And I think this data is from Lola, actually. You can combine these techniques. Here we're using CFSEDA and cell trace violet with immunophenotyping and are able to distinguish the individual populations in cells that are expressing CD4. So you can combine these techniques with surface immunolabeling very easily. And this is very commonly done to look at subset-specific proliferation. Now, I will not go through these charts in any detail. These are from Kylie. I'm including them because you'll be getting the slide. But these techniques are not easy. They do require a lot of optimization, and you need to spend a lot of time getting them right. You will not get them right the first time. You have to titer the concentration, the incubation times, the type of dye you're using, but there are good troubleshooting guides for both the protein dyes and the membrane dyes. And these are in the literature, and I advise you follow them. Now, as I mentioned earlier, we can get a lot of information out of these techniques. We can calculate precursor frequency, proliferation index, because as we mentioned earlier, proliferation is a stochastic process. If you stimulate 100 cells, not all 100 cells are going to proliferate, or some may proliferate immediately and some may proliferate in 4 hours. So out of a parent population, by analyzing these peaks, we can estimate the number of original precursors. And in this little cartoon example here, only about 10% of our stimulated population actually go on to produce the 4th, 5th, and 6th generation. A lot of our cells are not dividing at all. And in fact, you know, even the first the first few generations as well, they ultimately, when this population was analyzed, very few of them were remaining. So using the curve fitting software that's available to us, we can get very detailed information about the complexity of cell cycle, much more than we would get from DNA content alone. I'll include another nice technique here. This is data from Lola, actually, using something called FUCI, fluorescence ubiquination-based cell cycle indicator. In this case, this is a cell line, and there have been transgenic mice developed for this as well, where that express fusion proteins associated with different phases of the cell cycle. CDT1, in this case, is associated with a red fluorescent protein, and H-geminin over here is associated with a green fluorescent protein. So based on the phase of the cell cycle, we will see different fluorescent protein expression from G1 to SG2, and also a period where both proteins are expressed. These are endogenous markers. We're not adding any reagent here. They're already being expressed in the cell line, in the cell line or in the mouse. And there are a number of different combinations out there. You know, we now have hundreds of fluorescent proteins available to us, and these different combinations can be used to allow us to monitor cell cycle phase based on fluorescent protein expression. This is a section of a mouse embryo where you're able to see green, red, and yellow expression depending on where cell proliferation is occurring in the embryo at the time. And that data can also be analyzed by looking, in this case, at green and red fluorescence expression as well. So we have tools out there available to us, and these are getting actually quite sophisticated. We now have 3, 4, 5 cell cycle associated proteins that can be looked at using multiple fluorescent proteins. And Lola can tell you more about these experiments. But these are very interesting tools for monitoring cell cycle progression. So this is what we've talked about. We've looked at DNA cell cycle, immunolabeling, thymidine, analog incorporation, dye dilution assays, and fluorescent based cell cycle biosensors. But as we all know, cell growth is complicated, and we should never just look at one thing. So this concept of cell health is now becoming more prevalent, where we don't just look at cell proliferation, but we look at apoptosis, we look at cell stress. There are many aspects of cell physiology that we can look at, and we'll be hearing about some of these, I think, in the rest of the workshop. But we have many fluorescent tools for doing this. And I don't think anyone should just look at proliferation, because proliferation is usually occurring with a backdrop of apoptosis or senescence or autophagy, other processes that are going on. As many of you know, we have wonderful tools out there for looking at apoptosis. Again, this is something that we started looking at very early in the history of flow cytometry. Most of you know about gene-directed or signal transduction-directed cell death, where a tissue will intentionally kill off unwanted or dysfunctional cells. The cells shrink, the DNA becomes chewed up. We have expression of enzymes like caspases, activation of certain signal transduction markers. So I'm sure many of you are familiar with some of the assays out there. We can use low concentrations of DNA dyes just to look at whether the cells are dead or not. Many of us combine these assays with our phenotyping. An XN5 is a very common method for looking at cell death. There are fluorogenic caspase substrates out there that we can use to look at cell death. Caspases are enzymes that are commonly expressed during apoptosis, and we can combine these assays together to get a very rich picture of the cell death process. And apoptosis and proliferation often go hand in hand. We can look at mitochondrial function. There are many mitochondrial dyes out there that many of them measure the inner membrane potential, roughly a measure of mitochondrial function, of ATP and energy production. There are also mitochondrial dyes out there that are potential neutral, but allow us to count the mitochondria. This is becoming an important concept in immune cell physiology, as cells can use processes like mitophagy to upregulate or downregulate the number of mitochondria in a cell. And there are dyes like NAO and TMRM that are not potential sensitive, but allow us to see increases or decreases in the number of mitochondria. We can look at enzyme loss. These are very old assays, but these were the original cell death assays, where enzymes would leak out of the cell and were rough measures of cell function based on enzyme loss. We can look at plasma membrane potential in the outer plasma membrane. There are a lot of dyes. They look very similar to the PKH dyes. They have these long lipophilic chains that will insert into the plasma membrane and will increase or decrease or alter their spectral properties based on the potential of the plasma membrane. DIO C63 is probably the most common one. There are fast-acting, short-acting ones. There's a lot of sophisticated chemistry being done here. We can look at intracellular pH, like mitochondrial number. This is for people doing, if you're doing seahorse, if you're doing any of these techniques where we're looking at cell function and physiology, overall cell function and physiology, intracellular pH, inside and outside different organelles is becoming, we're finding out, very important in cell regulation. And there are some nice markers out there. We can look at ion flux measurement, potassium, sodium, calcium. These are not new assays. They've been around for a while. There are also fluorogenic substrates for enzyme activity. The caspase assays are one of them for cell death, but you can look at aldehyde dehydrogenase, alkaline and acid phosphatases. Aldehyde dehydrogenases are expressed in stem cells for some reason. We don't know why, but they are. So we have fluorogenic substrates for looking at enzyme function. There are biosensors and FRET probes where we can look at these as well. We can look at oxidative phosphorylation. I don't know if anybody does MTT assays anymore. It's a calorimetric assay. I did them when I was a grad student in a microtiter plate. Basically, you're looking at ATP production. You're looking at whether the cell is generating energy or not. And there are fluorescent probes that will allow us to do that as well, that are used to both measure cell proliferation and apoptosis and overall cell function. So we have many, many tools out there that will allow us to look at these things. There are markers for cellular senescence. Beta-galactosidase is a common one, but there are antibodies that will bind to proteins associated with cell senescence. Autophagy, unfortunately, we don't have great cytometry assays for this yet, but this is a very important phenomena. When I started as a grad student, apoptosis was just becoming understood as being incredibly important to cellular and tissue homeostasis. Autophagy is in the same place now. We used to just think this was part of cell death. Now we realize that cells can downregulate their organelles, the number of organelles, or discard non-functional organelles as a regulatory process in response to environmental stress. We do have, for example, there's an LC3 GFP fusion protein out there. Currently, the techniques for looking at autophagy are mainly imaging by microscopy, EM, or fluorescence. But I'm hoping we're going to have some flow assays out there. I think the Nobel Prize for biology or for medicine about 7-8 years ago was given for autophagy because this is turning out to be an incredibly important process. And hopefully we'll have some good techniques out there for that as well. And I'll just end with the fact that if you're doing cell cycle analysis, there are, I would say, probably thousands of potential protein targets you can look at simultaneously with DNA. This is actually this is a paper also from Dr. Darzenkevich's lab. He did cool stuff. He decided to look at tobacco and nicotine-free cigarettes to see if they were truly safer than normal tobacco. He was, again, labeling for 2 DNA damage markers. You can see that DNA damage occurs during exposure to tobacco, not surprising. But then he looked at vents. He looked at vapors over here, and they're actually worse. They cause even more cell damage than tobacco does. So he knew all the way back in 2010 that vaping is a terrible idea. And we're still trying to figure that out now. But these are the types of tools you can employ to look at what's going on in the cell, not just cell cycle, but overall cell physiology as well. So I will end there. As I mentioned, this talk actually came out of a CytoU proliferation education module that is online, that is on the ISAC website, that Kylie, Paul Wallace, and Kathy Muirhead over here designed. And the data really has come from a variety of places. Just to give you a few names here, Bruce Bagwell developed the ModFit software. Bruce is one of the inventors of compensation. You can thank him or curse him, depending on how you feel about compensation. But Bruce wrote all the mathematics for doing compensation analysis. He's really one of the founders of flow cytometry. And we have a variety of other people here who work in this area who all contributed to this presentation. So I think I will, with 5 minutes to spare, I will stop there, and we can, if we have time for questions.
Thank you very much, Bill. Wonderful presentation. Before I introduce Bill, this morning, as you may realize, we will switch to all different type of application of flow cytometry. So this was the first part of cell cycle proliferation and all these different type of assays. Then we will move to two different talks of other applications. Any questions? Questions?
Yes.
Hello, Will. Very nice talk.
Oh, thank you.
I want to know which is a good control of apoptosis. If I want to measure apoptosis, I want to be sure.
Absolutely, yeah. That's it. We didn't talk much about apoptosis today, but it's like any other technique. You want to have, if you have a technique, you want to have both a negative control and a positive control. You want to control where the cells are not dying, and you want to cells where the cells are dying. So certainly for a positive control, if for example, you're looking at a cell line, a tumor line, and you're testing drugs, You want a drug that's going to kill the cells as a positive control. There are different ways to do this. There are drugs like staurosporin out there that are toxic 10 different ways. They seem to be pretty non-specific. They seem to kill everything. So we have used those for positive controls. Some people, and even some commercial kits, do things like freezing the cells. They cause disruption of the cell to produce a positive signal. I don't like to do that because it's not signal transduction mediated. It's really necrotic death. And I'm not a fan of that. I would much rather you find some type of signaling mediated inducer. And that can be hard because a lot of these, like tumor lines, exist because they don't like to die. You know, they're drug resistant. So it can be a challenge. We have used drugs. We have used radiation. Hypothermia can often induce apoptosis. We often don't like to incubate our cell, keep our cells at 4 degrees because it turns out the EL4 cells I showed you here, if you put them at 4 degrees, they immediately start dying. They don't like to be cold. And this is an apoptotic response. It's a stress response. So you certainly want to find a positive, some type of positive control and also a negative control. One situation we often encounter too when we're doing primary cells is that there will often be a background amount of cell death, 5–10%, that's always going on. So your negative, your times 0, might still have some dead cells in it, and you need to take that into account as well. Is that what you're thinking about control?
Yes,
or
because I was thinking that cells die like in different mechanisms.
Yes.
So I want to be sure that the mechanisms in my positive is apoptosis and not
Absolutely, sure. We also have, like the EL4 system I showed you, we sort of use that as a test of our systems, even if that's not what we're doing our research on. It's a known system where we can make sure our assay is working, even if that's not what we're measuring. But some cells are very stubborn. Primary, human PBMCs, if they're not stimulated, they don't want to die. They're not cycling. We've used drugs like cyclohexamide, actinomycin B. Anything that blocks the cell cycle will generally kill a cycling cell. But if the cell is sitting in G0, they don't necessarily want to undergo apoptosis. There's no real stimulus for it. So it can be a little hard sometimes to find a control. But we've used staurosporin, actinomycin D, cyclohexamide, anything that will disrupt the cell rather badly can be used, certainly. Serum deprivation can do it, although serum deprivation can also induce autophagy. It doesn't necessarily kill the cell. The cell just becomes quiescent and starts to downregulate its energy requirements. But I think that's an important part of designing any apoptosis assay. But I'm not a fan of just freezing the cells or, disrupting them in some way, because that's not really apoptosis. We also have the challenge now, too, that there are many types of cell death, like necroptosis. There are other mechanisms that are not necessarily dependent on the same signal transduction pathways that classical kind of immune cell apoptosis is. In necroptosis, caspase 3, which is very dominant in other death systems, isn't present. It uses different caspases, different mechanisms. And in those cases, we're often more reliant on looking for the signal markers like RIP3 kinase and other things associated with that type of death.
Okay,
But we can talk more about it if you want, because that's something we do a lot of in our lab.
Thank you.
Sure. Okay.
Thank you very much, Bill.
Thank you.
Now we will switch to Sophie. Very clear presentation.
Thank you. Yep.
Hi, Sophie.
Hola.
Hola. ¿Cómo estás?
Muy bien, muy bien.
We are sharing your screen on the big.
Yes, so I'm trying to remove this. Okay. Wait.
No, volvamos.
We saw
Okay.
Sophie on the screen, but not there. Just 1 second, Sophie.
Yeah, no problem.
Just a couple of seconds, Sophie, because we can see you on the Zoom and YouTube. We cannot see here in the room.
Okay.
In the meantime, I etiqueta. Aznares. No. Difficult. Everybody knows who name is. This was on the fusion yesterday. If any of you forgot.
Yes, probably it's me.
Next time you will have one.
Yeah, probably. I will be there for sure.
Yes. Now. What's happening? Okay.
Ça va?
Ça va bien.
Oh, so I can do in French, right?
No.
Are you sure?
Maybe Sosha will stay. So we will continue now with the second part of the applications part of the talk. And so Sophie now will deliver a talk on nanocytometry. Thank you for your patience.
Oh, thank you very much. So the idea of this presentation, which is a short presentation, is really to introduce what we call nanocytometry. Maybe you probably have seen already this type of representation in which we can see clearly what is the area and the range of detection of what we call conventional flow. Is it okay? Yeah. Conventional flow, in which we can see the boundary of the detection of conventional cytometry. And you know that roughly we consider that as soon as you have a saline filter on any instrument, this was the smallest limitation in terms of detection of particles. So you know that on any instrument, there is a saline filter of a 0.2 micrometer. And so this was the limit. This is the limit of conventional flow, knowing that below we are completely, let's say, contaminated by the background and the electronic background, the fluidic background. And so since recently, I will show you, there is a new area in our field that engage the capacity to use flow cytometry to detect viruses and extravesicular particles. And you can see here that you have also a representation of the scattering and the size of the particle. And we can see that exosomes, Zika, adenovirus, SARS-CoV, etc., most of the pathogens that we have also in the campus of Pasteur. You know that we are involved in a lot of infectious disease studies. So this become more and more interesting to be able to detect. And so just a rapid overview of what we call extravesicular vesicle. So it's really an umbrella term to define microvesicles, exosomes, microparticles, and viruses. And so for a long period of time, those elements were considered as cellular debris. Okay. We know that most of the cell secreted this type of small particle. And there is an action from the cells, knowing that they remove some proteins, some cytotoxic agents, and it becomes so a metabolic way to remove the dust and the waste. On the other hand, what has been described quite recently is that those vesicles contain a phospholipid membrane, and that into these vesicles there is some genetic material. And so from that part, those vesicles, those extravesicles, those EVs, become interesting because they have a role in cell signaling and in the molecular communication. And so this was the first point. The second point was the description of those EVs in the term of those EVs in the application of some diseases. And so we know that you can have a modulation of those EVs production depending on a certain pathology. So it has been demonstrated in obesity, in diabetes, in cancer, in cardiovascular disease. And so they become really interesting in terms of biomarker to follow the status of the patient or to follow some treatment. And so clearly we have some example here demonstrated that those EVs can become a third way for the cell-to-cell communication, intracellular communication, knowing that they are everywhere. So this is an example of the SARS-CoV-2 that was in our life and still again. And you see that in patients that were affected by SARS-CoV-2, into the cells that are infected by the virus, there is a production of exosomes, and those exosomes has been found in the blood circulation, and they have been, some group have been able to detect them as a biomarker to, let's say, anticipate a specific signature that can help the physician to understand the physiopathology of the long COVID, for example, with metabolic syndrome, with inflammation and diabetes. And so this is an example just to show you how those small particles can have also an impact both as a biomarker or as a therapeutic target, so really for translational potential. And so one of the most advanced area is probably to use those EVs in urine to detect prostate cancer. It's also really interesting in terms of all the pathology of atherosclerosis and cardiologic issue. So you see, there is a real development and a real interest. And so those are the technology that are used to explore the EVs and the small viruses. And so for sure, the gold standard reference is the electronic microscopy. So we all know that it's not super easy to go to the electronic microscope. And you cannot do a large screening, or it's not possible to do high throughput with this type of instrumentation. And so for those who are involved in EVs since a long period of time, you have this technology that we call the nano tracking analysis. And so the nano tracking analysis is based on is tracking the Brownian motion of those particles, and is able to detect the size and the quantification of those elements. The second, so the name of this instrument is DNTA. And the second important instrumentation is the RPS, the Q nano. And in that way, we will evaluate the distribution in terms of size by the resistive pulse that is caused by the particle that is passing through a pore. So this is the three of the technology that are present in EV's lab and electronic microscopy. And more, flow cytometry, so conventional flow, has been adapted to detect those small elements. It could be classical flow cytometry. It could be also by using some beads. So, you can use beads that are coated with antibody. And you trap the EVs, and you can do a sandwich ELISA staining in a certain way. Like that, you can define the intensity and the specificity of some molecules. So maybe this is really adapted to exosome because we know that they express some tetraspanin. It could be also a way to use the imaging cytometry. And so we have also the capacity to cell sort those elements as soon as we have fluorescence. And there is some application in analysis by using conventional systems. So this is the Cytoflex from Beckman Coulter. And there is a specific small analyzer that is now available on the market, really dedicated to those small particles. And so the aim of my presentation will be to show you different examples of how flow cytometry in a general way, so I mean all those different instruments, has been involved to provide support of EV's project. This is interesting because it was the projection of what was using in terms of material and technology for the study of small particles. And you see that this has been published in 2016. And so you see that flow cytometry was really in the middle position of all of them. I can tell you that today probably flow is on the first or second position in the different lab. Why are we motivated as cytometrists to engage ourselves into this area? So we know that flow cytometry can differentiate different object populations. So we know how to differentiate heterogeneous population. And we know that those small particles, those subcellular particles, don't generate the same size object. And so you see, roughly, there is object from 40 nanometer to 1 micrometer. And so with flow cytometry, we can have an idea of the size distribution and we can measure the concentration. So really, this is to show you how those particles can be detected. So you see in fluorescence or in electronic microscopy. And this is here a representation of this heterogenic distribution in terms of size. So really size does matter. And just to show you what we have been able to manipulate locally here. So exosomes is 150 nanometer, microvesicles, apoptotic bodies. So you see HIV, Zika, which is really small, SARS-CoV-2, EVs from fungi, from bacteria, and from archaea bacteria. And so really to illustrate this growing interest in our field is really to show you the number of publications that combine EV's detection by flow cytometry. And you see that from nothing to the up, from the beginning of this century. And so mainly is due to the fact that we have been able to correlate the side scattering with the diameter of the particle. So using the side scatter to be able to define the size of the particle, being able to trigger optimal triggering on cytometer. And we have the chance now to have specific beads that are silicate beads that have the same refractive index as the real particle themselves. So this is still ongoing discussion in this field, but having the capacity to say, okay, if I define this area corresponding to a certain size of beads, it will correspond to real biological material. And so the idea to use forward and side scatter, and mainly the side scatter, to measure those small elements as well as having fluorescence is really interesting. And you know that now we can go below what is conventionally done by flow cytometry, meaning that we can go to exosome targeting, we can go to virus targeting. There is a limitation, a real limitation, because this is the flow cell. And this is the laser beam. And you see that when we do the interrogation point and when we inject into our instrument a certain number of vesicles, so concentration of vesicles, in this small section here, this small volume, 54 picoliter, it could represent 800 vesicles. And we know that our aim when we do flow cytometry is to interrogate every single particle one by one. And so this was really a limitation. And this is why the forward scatter is difficult to be used. And this is why for cell sorting, I will show you some example later on, a specific strategy need to be put in place. So this is the first example done with beads. You see that here you have the representation of the size of the beads. So the red are 100 and the green 160, etc., etc. How to do so? So this has been done on the Cytoflex, which is a conventional instrument. The only variation is the fact that we can use the violet laser side scatter, knowing that the SSC diffraction on the side is not coming anymore from the blue, but is coming from the violet, so the 409 nanometer. And we have also the capacity to have the forward scatter with a PMT. And so you see that there is here absolutely needed to do a thresholding on the fluorescence. Those beads are positive in they are positive in the FITC channel, so they are positive in the green. And you see that if you don't have the capacity to put this threshold, it will be really, really super difficult to detect them because we are completely dive into the electronic and the fluidic background. So this is roughly the basic approach. To detect those small particles, you should have a fluorescence readout to be able to do a threshold, to place a threshold, and to see the side scatter, not on the blue, but on the violet. So this is another example, which is, let's say, a real application, still done on the Cytoflex. And in that case, we are looking to EVs that are produced by platelets. And you know that on the platelets and on their EVs, we can use some markers. So here is the CD61, which is a specific marker for the platelets and for the EVs. And so you have the different controls. So the CD61 versus the side scatter violet. This is the control with just the antibodies. This is the whole blood. And so we have EVs in the whole blood, but we have also platelets. They are there. You can see that we have a direct detection on the plasma. And as soon as the plasma has been enriched and filtered, you see that we can enrich this fraction. So this was really to show you that from blood samples from plasma, we can absolutely, with a super easy way, detect those small elements. Another application, this is what I was presenting in the general slide, is the fact that we can target exosomes still by using the multiplexing approach. So you know the principle of the multiplexing. You use beads, and those beads are covered with capture antibody. So the cocktail of the capture antibody could be specific to the EVs that we want to target. In that case, we are looking at to catch exosomes, and you see that it's a kit that is specific for the 61, the 9, the CD9, and the CD81. Those are testraspan. Those molecules are specific of exosomes. And you see that after the incubation, we have this element. So we have a huge bead that is covered by the exosomes that are become fluorescent. And so from the forward and side scatter, we target the bit size, and we have here the capacity to follow the PE. It will be the different specificity, and you see that we can follow the shift in the APC channel of the expression of the tetraspan. So this is, again, a real application around the exosome. You still understand that in that case, we have fluorescence, okay? We were talking about the image stream. So this is an example of imaging cytometry. So you know that the image stream has the specificity to have a spatial resolution, which is not at all what we can propose in classical flow cytometry approach. And so I would like to show you some example also with beads. You know that it works like a microscope, so you have a magnification possibility. So you see that the highest magnification is 60x, and you see that we can detect a small element. So again, an example, how to detect single particles that are fluorescent, still fluorescent. And so you see, this is a cocktail of beads. So those one are the 100 nanometer. P3 will be 300, 500, and here you have 900 nanometer. And so if we target those this gate, and we can see that it's quite difficult to see some fluorescence, but still we see the small beads. Difficult to see the side scatter and difficult to see the bright field. But in any case, we can detect those small bits, and you see that the correlation in size is nice. This is the 300 nanometer. This is the 500 nanometer, and finally the 900 nanometer. So this is a way to show you that based on the fluorescence, with a side scatter that is quite efficient, and we can use the one from the violet, as soon as you have a fluorescence readout, then you can detect them on a really easy way. As soon as the instrument is really clean, etc., etc., we need to have a really cleaning procedure that allows the detection of those elements. If we talk about cell sorting now, cell sorting is a little bit different. What we have trying to do locally, and so this is my experience in my hand. So we have trying to use square cuvette instruments, like the FACS area, the FACS fusion that we were talking about yesterday. So you know the interrogation point is really into the cuvette, so on the quartz cuvette. You remember that I explained you that it's difficult at that time, at that interrogation point, to target only one element. I believe that also due to the fact that we have the drop delay, that it's far away, and due to the fact that maybe we have turbulence, it was really, really difficult in our hand to be able to sort those particles, even if we have fluorescence. Meaning that the strategy to use cell sorter with cuvette-based was really for us a limitation. And so we have trying to do the same on the Jetiner system, and in that case it was really well working. Just to provide more information on the FACS area fusion family, we were able to see the beads, to see the particle fluorescence, but finally in the collection tube we have an efficiency of sorting that was really poor, which was absolutely not the case in the Jetiner family. So here again, just to show you what was our strategy. So we take beads, fluorescence beads, here on the FITC, on the GFP parameter, with the side scatter. In our hand, the best scattering thresholding was on the yellow-green. So this is a specificity of this machine. We can apply a side scatter on every laser, and it was with the yellow-green that it was the nicest. And so you see that we were really well able to define 0.16 from 0.2, from 0.24, from 0.5. And so from that point, we started to do a real application and a virometric assay. And so this was a project that we have developed with the virology department. And so they have produced GFP particles. Then, so it's here you have the transfection, so VPR GFP. And so after ultracentrifugation to have some concentrated particle, we have done some analysis on our cell sorter. And so what we have done here is to be able to detect GFP signal versus the side scatter. And for sure, we have put a threshold on the side scatter of the yellow-green. And we were able to demonstrate that we have 3 gates, so the low expression GFP, the med, and the high. And we have been able not only to detect, but we have been able to sort them. So this is what we have done. So this is the post-sort analysis. We have control that we have viruses in our different fraction by measuring the P24 protein by ultrasensitive ELISA, and confirm that by confocal microscopy, that the low fraction, the med fraction, and the high fraction correspond to 3 different gates. And you see that finally, what we call the high GFP was finally the fact that we have an aggregate of small particles and of the viruses. And the idea in the project for our collaborator was to study the capacity and the infectivity of the HIV virus in those 3 different strategies, so single viruses or viruses that are aggregated. And so they demonstrate in these publications that there is a link between aggregation and infectivity. So from that part, we also developed, so not only the viruses cell sorting, but also the EV cell sorting. And so this is an example that we have also implemented. So we are working on fungal EVs coming from a pathogen that is the Cryptococcus neoformans, which is involved in chronic meningitis and that will develop a pulmonary and cutaneous infection. We know that those small EVs are covered by a sugar, by a polysaccharide. And so we have used fluorescence because we need fluorescence to be able to cell sort. So we have stained those EVs with an anti-GXM to be able to detect them. So this is the wild type. And this is here a mutant in which they didn't express this polysaccharide. So here again, we have been able to cell sort and then to go to the electronic microscopy. And from that part, we have been able to demonstrate that at the surface of those EVs, there was some that were carrying some decoration, and some other EVs didn't carry those decorations. So this needs to be deeper study. And from that element, the team that we were working with, they were interested of screening their batch of production of EVs to know if there is a modulation in terms of production of those small particles in different culture conditions, with or without antibiotic, with or without, and the timing of production of those EVs during a time courses. And so we were using our sorter, not for sorting, but for doing the analysis of the batch that were produced. And so this is an example. So in that case, we were not using this antibody, but another one. It was a lectin Kona GFP. So this is the PBS control, this is the wild type. And here we were able to say, okay, we can target correctly the particle. We were using the TrueCount tubes to be able to say, okay, we have a certain amount of particle on it. So we were able to do detection and quantification, but it was a little bit pity to use a cell sorter to do so. And so we were looking to a new technology. And so, as I said, the NTA, so the nanosite, is still the gold standard reference in the laboratory. But with this instrument, you don't have fluorescence. There is the cryo-EM. We know that we cannot use the cryo-EM for batch screening. And so there is on the market a new instrumentation that has been launched quite recently, which is the NanoFOCEM, which is a cytometer that can provide, that is really specific, that can provide information on size distribution. And we have correlate that with this particle coming from Cryptococcus, we have roughly the same size at what has been observed in cryo-EM. And so from that point, we have requested some budget, and we have purchased this small analyzer. So you have here the representation. So you see, it's still the same principle. You inject your sample. So what is really specific is that the sample line is really short. So the pathway in between your sample and the flow cell is really short. The sample core is also specific. We have a specific detected volume. 20 nanoliter is uptake. So you see that we inject on a really small way the EV. So we increase the time of in front of the interrogation point to have the maximum capacity of detection. It's a really basic instrument. There is only one laser. The detector are ADPs with a side scatter and with the possibility to have also some fluorescence redundant. And so it's really simple. We can detect elements below 100 nanometer. We trigger on the side scatter of the blue laser, and we can detect a 40 nanometer particle. So it works with a really small flow rate, with a specific to have a better focus and a better interrogation point. And the system requires 100 microliter, and the acquisition is done for a long time. It's 1 minute acquisition. And so this is what we have been able to do. So still on the same cryptococcus EV, so you see that we have demonstrated and we can calculate with beads that are standard for us. We can define the size of those particles. So we have an information regarding the relative size. And because our collaborators have generated mutant, and we know that the production can be modulated. So we have demonstrated that we have the wild type production, and within the different mutants, we have a decrease in the production. So we can calculate relative concentration of the different mutant production. And we have put in correlation the time of growth, the time culture with the production of those EVs. And so having a time courses of EV production. So this was the basis of the test. You can have a time courses. You can module. You can add in your culture different agent to, as I said, antibiotic, antifungal element, and you can modulate the production in the different culture media. Another example with a virus example. So this is the SARS-CoV-2. So the SARS-CoV-2 is 70 nanometer. Here again, we have roughly the size of the SARS-CoV-2 that is produced in the supernatant of viral cells that has been infected during 30 hours. So we have a size estimation. And here again, we have been able to demonstrate in parallel the cell growth. This is the time post-infection with the production of EVs that can be detected in the supernatant. So here again, we can propose to the virologist a way to, to qualify the batch production of those small particles. For sure, exosome is the most easiest way to use any approach of nano and subcellular element project. In that case, we are on murine exosomes. Here again, we have used the CD63 and the CD9. And you see that we have here the capacity to define a size. This is the control of double negative for both color. This is the single color for the fits. This is the single color for the PE, and this is the double positive. So this is roughly what we have been able to show locally, but this is something that is developing a lot. And so just to show you that there is a network and a working group in between flow cytometry, so ISAC, with the ESV, which is the International Society of EVs, with also the Society of Thrombosis and Hemostasis, because there is a lot of application in that field. And we are personally engaged in the French local workshop with both society. And you know that at ISAC, and probably people from ISAC can confirm that there is some thematic strong connection in between ISAC and those topics. I would like to say that there is something that is really important in nanocytometry that we didn't have in hand. I know that, for example, and Zosia can explain that to you. There is a cancer center in Paris that is named Cure Institute, in which they have created a dedicated platform for the detection of small EVs. For sure, they use cytometry. nanocytometry. But what is really nice in their hand is that they will also monitor the production of EVs, meaning that the production is something, but I didn't have the end on the cleanup part, meaning that do we need to do ultracentrifugation? Do we need to do size exclusion column, etc., etc.? And we know that the preparation of the sample is super important. So this is something that we don't monitor for the moment. But what I can say is that today we have the nano FCM for the size and the concentration. We request an additional technology, which is a microscope. So the limitation of the microscope is still, let's say, 100 nanometer, but this will be a way also to characterize from a single drop of a preparation of virus or bacteria, of EV, the concentration. So there is no readout of fluorescence. So both of them will help us to do a size definition and concentration, and still have so the Astrios and the Bigfoot, so meaning the jet inert technology, to be able to cell sort them for further characterization. So as I said, electronic microscopy, but also mass spec for sure for the component of those different elements. And so I think I'm over my time, but just to say that this project has been is really in the hand of one of our engineers, which is Pierre-Henri Commer. He is the project leader of the Nanoflow in our facility, and all our collaborators that have been involved in this project and those different projects. And because it's my last time with you during this week, I would like to thank again the organizer for your invitation, for your the possibility to be part of your week of training. And so once again, I would like to thank you very much for your invitation and your attention.
Thank you very much, Sophie. Any questions in the audience or in the... Yes, one.
Thank you, Sophie. Sorry if I didn't get it. Did you do sorting using area? And
No.
if so, sorry. Yeah.
So exactly. So what we have done is that we are trying to use the area system to do the characterization of the viruses and the EVs. We were able to see really well the different particles, but it was impossible to have an efficient sorting. I believe that something is happening into the cuvette. I don't know if it's turbulence or whatever, but the system was able to see properly the small element, but we didn't have a correct recovery of those particles in the Eppendorf tube in the collecting fraction.
Okay.
And so we shift from the square cuvette to the jet inert system. And when you talk within the community of flow cytometrists, most of the people who work with small particles and want to cell sort, except the megavirus, for example, or large particle, it's really difficult. It's really difficult.
Thank you.
And I precise, as soon as you want to cell sort small elements, you should have fluorescence. Without fluorescence, we are blind, completely blind. The only instrument that is able to provide any results without fluorescence is this small cytometer here. This one and this one are not related to fluorescence readout. But as soon as you will use the image stream, as soon as you use the Cytoflex, or other, I know that the Aurora is also really good in the detection of small elements. So this is something that can be also developed by Maria. I know that the readout of fluorescence is really needed.
Thank you very much. Any other questions? If not the case, we thanks again, Sophie, for really, your contribution were very nice during this. Unfortunately, not in person, but we hope that next time you will fly from the Tower
Yeah.
Eiffel to Montevideo, and you
And
have
you see
some
you
coffee breaks and meets with us.
and you can come to Europe. And like I said, if one day you want to you're in Paris and you want to visit us, you are more than welcome.
Yeah, you know that here there are almost 30 or 40 people, so all the people are invited as well.
Exactly.
Yes.
We can do a
Thank you.
group. We can do a
Yes.
group.
Thank you very
Thank
much.
you very much. Thank you. Bye.
So now it's time for a coffee break. We will meet again at 11 o'clock.
Montevideo, dentro de Montevideo 2023. La que está en PDF.
No, el PowerPoint.
El último. Esta. Está. Control C.
Tiene el mismo nombre que le habíamos dado antes.
Y esa habían quedado acá. Es la de application. La deleteo y pegamos la. Perfecto. La abrimos. Perfecto. Y ponemos share screen. Con screen 2. Pero no me pone la screen 2. Pero anda tranquila que veo esto, y si no lo llamo a Sebastián. ¿Cómo está?
¿Te diste cuenta que algo me había pasado?
¿Estaba muerta?
Ojalá esté razonando en irse.
No, esperarte esperé un minuto. O sea, es esperar. Esperate, esperé un minuto.
¿Qué sentí? Muerto. ¿Qué sentí? Y empezó a bajar la presión, ¿qué pasa?
Sí, estaba bajando a cero, vas a contar.
Y empecé con todo y dije, traje. Bueno, y Gabriel hizo la verdad que la paciencia.
Pero vos tenés que fijarte en esto. Está buenísimo, qué bueno.
Vamos muy bien, trabajaron bien. ¿Qué así te iba bailando? No, bailaste hoy.
Ahora cantamos.
Cantamos.
No.
Pero te envié les envié
Pues de la Macarena lo vimos.
No. No has visto el otro.
Vimos que estabas bailando bien.
Che, voy a parar.
Bueno, vamos antes de empezar con Florencia, su segunda charla, les quiero pedir un favor que antes de ir a almorzar, es decir, va a venir la charla de Florencia, van a estar hipoglucémicos, pero igual se dirigen al laboratorio informático y se loguean en las computadoras y ya dejan abierto el Flow Show. Cosa que después podemos ir tranquilos a almorzar y en el postprandial, que estamos todos dormidos, ya tenemos las licencias corriendo. Y después para la parte práctica nos vamos a dividir, ya les voy a dar las indicaciones, pero les pido por favor que aquellos que ya les ha funcionado la licencia, antes de ir a almorzar ya lo dejen activo en alguna computadora. Y aquellos que no han podido loguearse, nos lo digan, porque capaz que pueden usar en el segundo turno, pueden intercambiar, es decir, uno que se loguea en el primer turno y después va al equipo, alguien que estuvo en el equipo puede seguir usando esa computadora siempre y cuando no se deslogue. ¿Está bien? Bueno, pasamos a la tercera charla de aplicaciones a cargo de Florencia. Pasamos desde ciclo celular, proliferación y estado saludable o no saludable de células a la nanoescala. Y ahora vamos a ver con qué nos sorprende Florencia.
¿Se escucha? A ver, probemos si se escucha. Hola. Perfecto, buenísimo. Hola, buenos días a todos. Muchas gracias nuevamente por esta invitación. La verdad que está siendo un curso súper intenso, pero me parece que todos lo estamos aprovechando, tanto los alumnos como los teachers, porque me parece que estamos creciendo todos. Así que Mariela se me fue, pero realmente agradezco a Mariela y al equipo por haberme invitado, porque realmente estar con semejantes monstruos de la citometría es un honor, en el buen sentido. No, escucho historias de Soya, de Bill, de las chicas, cosas maravillosas. Realmente es un placer estar acá con ustedes y cantando con ustedes. Bueno, ya es bueno, mi charla es, como ustedes saben, yo soy científica, no soy citometrista de full time, ¿sí? Entonces mi charla va a estar más enfocada a cómo uno utiliza la citometría de flujo en el contexto de un proyecto de investigación, ¿sí? Vamos a explorar diferentes estrategias experimentales, pero con el enfoque de contestar una pregunta, ¿sí? En el contexto de una pregunta, de un objetivo. Y más allá de todo lo que estamos viendo, que son millones de técnicas, millones de cuestiones inherentes a la citometría de flujo, profundizar en el concepto de la versatilidad que tiene la citometría de flujo para adaptarla a lo que nosotros necesitamos y contestar nuestras preguntas específicas. Y de esa manera se lleven ideas, quiero que ese sea mi pequeño aporte hoy, que se lleven ideas como para implementar en sus proyectos, en sus laboratorios o charlar con sus colegas. Les pido disculpas por adelantado porque yo soy inmunóloga, con lo cual les voy a hablar 100% de inmunología. No hago plantas ni bacterias, que hubiera estado bueno quizás, pero como no tengo experiencia en eso, la realidad es que, bueno, les pido disculpas por eso. Va a estar, la charla va a estar, exacto, bien, sesgada para ese lado. Primero, algo súper básico, pero que a lo largo del tiempo he visto que mucha gente no le da mucha bolilla. Y es los cambios morfológicos que sufre una célula después de un tratamiento. Ustedes dicen, secundario, me voy a la fluorescencia, al multipanel de 20 colores, a cómo se expresa. Miren la morfología. O sea, yo a los chicos, a los becarios les digo, Vayan al laboratorio, miren las células, analicen, mírenlas al microscopio, vean cómo van cambiando en el tiempo. Y eso también lo podemos hacer en el citómetro de flujo. ¿Por qué? Porque los linfocitos quiescentes tienen una morfología, células pequeñas, poco citoplasma, poca complejidad, pero cuando uno las estimula cambian y cambian mucho. Yo estoy poniendo una estimulación antigénica con Mycobacterium tuberculosis, pero si pongo un mitógeno como PHA o cualquier otro, las células se convierten en gigantes. ¿Bien? Y eso yo lo tengo que controlar, lo tengo que ver, es un resultado. ¿Bien? Entonces, es muy pavote, es cierto, pero es muy importante que uno controle su experimento desde el punto de vista de los cambios morfológicos que puede llegar a ver. Y ahí avanzar en los cambios que yo busco, la pregunta que quiero contestar, esos marcadores terciarios que tanto nos habló María, que son maravillosos como para contestar preguntas. Pero siempre empezar por el principio. Bueno, Bill les contó maravillosamente cómo uno traquea la proliferación celular. Cuando uno ve en las publicaciones y en internet cómo desde una célula en estado basal se van viendo las diferentes hijas, bueno, los cuidados que uno tiene que tener, ¿no? Para que todas las células se marquen lo más uniformemente posible, yo pueda tener un pico bien angostito de estas células en estado basal para poder diferenciar después a las diferentes hijas, ¿no? La fluorescencia se va diluyendo a medida que las células se van dividiendo. Pero bueno, la vida real es diferente. Y acá les estoy mostrando resultados nuestros, donde hacemos estimulaciones con Mycobacterium tuberculosis y comparemos, ¿no? ¿Por qué una cosa y por qué la otra? Acá estamos teniendo todas las células proliferando en fase, todas juntitas. Madre, dos hijas, cuatro. ¿Bien? Acá no, porque acá, por ejemplo, lo que hicimos fue cocultivar células dendríticas cargadas con antígenos de Mycobacterium con linfocitos del mismo dador. Entonces, la activación y como decía Bill, es estocástico el evento, los eventos de proliferación, pero acá más todavía, porque uno tiene que esperar que la dendrítica procese, presente y active diferentes linfocitos que van a activarse con diferentes cinéticas, con lo cual la resultante va a ser esto. Nosotros después vimos, porque trabajamos con hormonas inmunomoduladoras, que la dehidroepiandrosterona incrementaba aún más esa proliferación, ¿no? Pero fíjense qué bodoque, que nada tiene que ver con esto, ¿no? Sin embargo, fue proliferación. Y acá lo mismo, este sistema es un cocultivo entre macrófagos, en este caso, infectados con Mycobacterium tuberculosis y cocultivados con linfocitos del mismo dador. Lo mismo, y acá, y esto lo hicimos con nuestro espectral, esto lo hicimos con el canto y esto lo hicimos con el espectral nuevo. Algo que me gustó del espectral es que finalmente alguito pudimos ver en las hijitas, nos dio la sensibilidad para ver eso, pero también mostrarles esto, los cambios morfológicos que se dan. Vuelvo a lo mismo, yo soy como repetitiva en este sentido, pero sacamos información de esto. Yo podría hacer un histograma y quedarme solamente con el análisis de la fluorescencia del CFC, de la proliferación. Pero lo que estoy agregando acá es un parámetro de morfología que me muestra que luego de la estimulación las células se convirtieron en blastos, ¿sí? Y que esos blastos son los que están proliferando. Entonces, fíjense que además de la información de la proliferación, estoy sacando información morfológica, combinando dos parámetros, un parámetro de fluorescencia y otro de morfología. ¿Bien? Así que bueno, como verán, la vida real, en mi caso, nunca llega a esto. Obviamente, si yo hago una estimulación con PHA de estas células, llego a este resultado, ¿no? Conseguimos picos angostos, o sea, tenemos buena resolución en CFC. Lo que pasa que después de semejante baile para las células, llegamos a este resultado, ¿sí? Otra de las técnicas que se usa muchísimo en citometría es el análisis de proteínas en líquidos biológicos, ¿sí? Que consiste en hacer como una ELISA sobre una perla. Yo tengo bits que están conjugadas con diferentes anticuerpos y que fíjense que tienen diferencias en su color, ¿no? Esta es más clarita, esta es más oscura, con lo cual tengo diferentes intensidades de fluorescencia para las bits. Cada intensidad de fluorescencia está coateada o conjugada a anticuerpos contra diferentes analitos, ¿sí? Pueden ser citoquinas, quemoquinas, proteínas, pueden ser, bueno, después se los voy a mostrar, pueden ser vesículas extracelulares. Uno lo incuba con la muestra, pone un segundo anticuerpo que puede o no estar conjugado directo o con una streptavidina, con un sistema de streptavidina, lo mismo con ELISA, pero sobre la vid. Se corre la muestra y después se analiza. Habíamos dicho que cada perla tiene una intensidad de fluorescencia, cada intensidad de fluorescencia, cada perla con una intensidad X está acoplada a un anticuerpo específico, con lo cual hago un ELISA sándwich y puedo tomar una fluorescencia X de perlas y hacer una curva de calibración con un estándar de concentración conocida para luego analizar mis muestras. Estos son datos de nuestro laboratorio donde analizamos citoquinas y quemoquinas humanas, ¿sí? Entonces ven que cada intensidad de perlitas analiza una citoquina o una quemoquina diferente y el corrimiento en PE, que es el anticuerpo que está cerrando el sándwichito, nos habla de la cantidad de analito que hay en la muestra. Sí. Micrófono. O si no, repito la pregunta.
Me surgió una duda en el volumen de muestra que requieres para hacer este ensayo, para poder obtener resultados, por ejemplo, de todas esas citoquinas. Por lo que mencionas una ELISA, uno tendría que hacer con el sobrenadante proteína por proteína, pero acá quizás entonces uno puede incluir varias perlas con diferentes anticuerpos y con la misma cantidad obtener un resultado con mucha más citoquinas, en este caso.
Exacto.
¿Qué cantidad de muestras?
Exacto. No me acuerdo cuánto, pero es lo mismo que una ELISA. Así. Sí, ahora sí, micrófono. Es muy chiquito el volumen. Acá Lola dice que lo hacen con lágrimas, que hay gente que lo hace en lágrimas, con lo cual el volumen es muy. Esa es una de las grandes ventajas que tiene, que vos podés analizar volúmenes muy pequeños de muestra. Bueno, en ese caso a vos te serviría para volúmenes muy pequeños. La desventaja que hemos visto en algunos casos, y en el caso de más de sobrenadantes quizás, es que si tenés alguna citoquina o quemoquina o algún analito que se te va de escala, o lo analizás con un ELISA convencional aparte, o repetís todo esto, que sabemos que es bastante costoso, con la muestra diluida como para que te entre el rango. Acá es lo mismo de siempre. Tenés que trabajar dentro del rango de linealidad de la técnica. Si se te va de escala, tenés que diluir tu muestra para que entre en escala. Con lo cual deberías, y ese es un trabajo que hacemos todos, investigar cómo es la concentración de tu analito en esas condiciones para ver con qué dilución trabajar en esta técnica. Toma, Lola. No, está bien.
No, que digo que siempre hacer una curva para tener idea dónde está, porque muchas veces lo que te pasa no solo que se te sale de escala, o sea, las curvas de calibración para estos kits tienen unos rangos de concentración, pero a veces tú estás en la parte baja, entonces tener concentraciones muy altas no te sirve de nada y lo que te interesa es tener más puntos en la curva en la parte baja. O sea, tener una idea de por dónde te mueves siempre es muy útil en estos ensayos.
Seguro,
No.
seguro. Siempre, siempre averiguar cómo va a ser tu concentración o tener un estimado de cómo va a ser la concentración de tus analitos en las muestras. Y esto para mostrarles cómo es la interpretación de resultados, que hay cada equipo, cada kit, cada empresa tiene su software de análisis, donde la curva se construye a partir de un estándar y uno entra con la intensidad media de fluorescencia de la muestra, uno no, el software, entra con la intensidad media de fluorescencia de su muestra y puede analizar su concentración. Es exactamente igual que un ELISA. Siguiendo con el tema de las bits, y no tenemos el mismo nivel que la charla anterior, ni los equipos ni el presupuesto, pero les quería mostrar de nuestro instituto, hay varios grupos que trabajan en IBIS, y una de las formas de analizarla fue haciendo una captura con bits couteadas con anti-CD63, se capturan las IBIS y ahí se puede analizar su fenotipo. ¿Bien? Entonces, en este caso se analizó CD20, CD81, CD107 y HLA-DR en sobrenadante de cultivo de una línea celular. ¿Está bien? Esto fue la puesta a punto. Este grupo trabaja en infecciones pulmonares pediátricas. Entonces ponen todo a punto sin utilizar muestras pediátricas, obviamente, porque son muestras súper preciosas. Así que bueno, saliendo de las perlas magnéticas y continuando con cómo uno encara una pregunta, ¿no? Y utiliza la citometría de flujo para contestarla. En este caso, también un grupo del instituto lo que quiso saber era si había alguna función citotóxica en la IgA de pacientes con HIV. ¿Bien? Si esa IgA tenía alguna función de citotoxicidad. Y si el tratamiento modulaba esa capacidad citotóxica de la IgA. Lo que hicieron fue un ensayo de citotoxicidad con un readout por citometría de flujo, donde unas células blanco, ¿sí? Esta línea celular, se coutea, se adorna toda con antígenos de HIV. ¿Bien? ¿Dónde estaba este chico de Perú? Ay, no, ¿dónde está? Allá. Él también lo está haciendo, pero para micobacterium, por eso. Entonces, tengo las células adornadas con el antígeno de HIV y se marcan con PKH26, que ustedes ya saben que es una sonda que marca membrana. Una vez tratadas las células y marcadas, se incuban con suero de estos pacientes, ¿sí? En realidad con suero tratado de manera de tener la IgA lo más pura posible, con lo cual en esta instancia el anticuerpo específico se va a pegar al antígeno de HIV y después se le agregan células mononucleares de sangre periférica de dadores normales que van a tener células NK. Se puede hacer con células mononucleares o se puede hacer con células NK purificadas. Las NK van a ejercer citotoxicidad celular dependiente de anticuerpos y van a inducir la muestra de la célula. Perdón, por último, estas células se marcan con CFC. Acá está, con CFC. Se marcan con PKH y con CFC, con los dos marcadores, ¿sí? El PKH va a estar en la membrana celular y el CFC va a entrar a la célula, ¿bien? Entonces, después del ensayo del cocultivo, se determina la muerte celular inducida por anticuerpos como el porcentaje de células PKH26 que perdieron la marca de CFC. Entonces, ¿cómo es? La membrana va a estar siempre marcada. Las células van a, porque yo tengo que diferenciar entre target y efectora. Entonces, el PKH me ayuda a identificar las células target. ¿Bien? Siempre van a estar positivas para PKH, pero el CFC en las células vivas va a ser bien intenso y cuando la célula muere, se generan poros y las proteínas marcadas con CFC se van a perder, van a salir. La resultante va a ser una baja de intensidad en CFC para aquellas células que murieron. ¿Bien? Entonces, es lo que vemos acá efectivamente. Las células cargadas con GP120 que murieron por ADCC van a tener menor intensidad de fluorescencia de CFCE comparado con el control, con células sin cargar. ¿Bien? Entonces, con una técnica que es relativamente sencilla, es bastante ingeniosa, uno puede determinar la citotoxicidad celular dependiente de anticuerpos. Antígeno específica, ¿no? Porque uno puede hacer ADCC, pero quizás no puede determinar esta función utilizando anticuerpos antígeno específicos. Esta técnica lo permite. Otro uso, que ustedes ya lo saben, obviamente, de la citometría multiparamétrica es el análisis de la funcionalidad de linfocitos, ¿no? Donde uno, después de un trabajo arduo, arma un panel de anticuerpos enfocado a analizar la funcionalidad de las células, ¿sí? Entonces, voy a utilizar anticuerpos contra interferón gamma, interleuquina 2, MIP1 beta, TNF-alfa y CD107A y B, que son dos moléculas que están en la membrana de los gránulos citotóxicos y que cuando la célula degranula, estas moléculas se expresan en la superficie de la célula. Bien, entonces es una molécula que me habla de la capacidad citotóxica de la célula. Sí, obvio. Nosotros lo que hacemos es estimulación antígeno específica y como control positivo utilizamos estimulación con PMA ionomicina. Vos lo que podés hacer es estudiar la respuesta global de la célula, sacándole la foto, digamos, induciendo la síntesis de todas las moléculas o la funcionalidad con PMA ionomicina. Pero nosotros lo que hacemos es estimulación antígeno específica siempre y utilizamos a la PMA ionomicina como control positivo. Pero sí, siempre tenés que tener. Sí, sí, porque ves, esto fue, sí, las ves perfecto, tal cual. Entonces, obviamente nuestra estrategia de gating, acá falta, no, está después. Son la discriminación de los dobletes. Ven que acá hay mucho doblete, porque como es una marcación intracelular, las células se pegotean. La determinación de la viabilidad. Acá hay un dump channel con CD14 y CD19. Miren los linfocitos, cómo están bien estimulados. CD3 y después se analizó en CD4 o en CD8, en este caso fue en CD8, se analizó cada uno de los parámetros. ¿Bien? Y después se analizó mediante Boolean gating, que si hay algún matemático me puede ayudar, pero básicamente es, yo le digo al software que me diga qué células tienen todas con el comando AND o qué célula tiene alguna de estas con el comando OR. Bien, eso es el Boolean gating. Entonces, puedo hacer análisis de este tipo, ¿sí? Donde puedo ver la respuesta a diferentes antígenos de HIV, las células CD8 que ejercen una función es o que produce una citoquina o que expresa el marcador de degranulación, 2 funciones, 3 funciones, 4 funciones o 5 funciones. ¿Bien? Este tipo de gráficos nosotros estamos súper habituados, lo analizamos con nuestros ANOVAs. Yo soy inmunóloga, no soy especialista en estadística, así que pido disculpas nuevamente, pero durante un tiempo decíamos, necesitamos algo que nos analice si hay diferencias en la distribución de poblaciones a lo largo de diferentes grupos experimentales, ¿sí? Este paper, Turk, que es una compañera del instituto, pero nosotros trabajamos en distribución de poblaciones de memoria, ¿sí? Y teníamos el mismo problema. Yo quería saber si había diferencias en la distribución de poblaciones de memoria entre mis pacientes con tuberculosis activa, tuberculosis latente, tuberculosis con HIV, bueno, demás. Y no lo encontraba hasta que nos cruzamos con este software que es gratuito del NIH, que se llama SPICE, por Simplified Presentation of Incredibly Complex Evaluations, que es sencillo de utilizar y que nos contesta la pregunta. Porque fíjense que la distribución de respuesta a NEF es diferente a la distribución de respuesta a P24 para un grupo determinado, ¿no? Estos son Delay Treatment Group para HIV. Entonces, con esta herramienta, no solamente puedo analizar datos multivariados, como lo veníamos haciendo habitualmente, sino podemos hacer un análisis más profundo y descubrir nueva información con respecto a nuestros datos. Y se puede hacer con datos de citometría de flujo y con otros datos también. Es bastante versátil. Nosotros lo usamos siempre con datos de citometría de flujo, pero para genómica y demás también lo podés utilizar. Así que bueno, quería mostrarles esto porque me parecía interesante que se lo lleven como herramienta para este tipo de análisis. Otro approach en el que estuvimos trabajando durante un tiempo fue ver cómo se transferían moléculas citotóxicas desde las células CD8 hacia el macrófago. Nosotros teníamos un proyecto donde queríamos ver, siempre en el contexto de la tuberculosis, ¿no? Cómo la diferenciación del macrófago hacía que sea más o menos susceptible a la citotoxicidad CD8 específica para Mycobacterium. Entonces hicimos estos settings experimentales donde a partir de PBMCs aislábamos monocitos, los diferenciábamos a macrófagos, a partir de ahí lo diferenciábamos nuevamente hacia macrófagos M1, M2, nos interesaba especialmente los diferenciados a M2 por IL-10, que es una citoquina que se levanta mucho durante la tuberculosis. Y una vez que estaban diferenciados los macrófagos, los cargábamos con Mycobacterium o con estos CEF, son péptidos de citomegalovirus, Epstein-Barr e influenza, que son como algo bastante estándar para ver citotoxicidad, ¿sí? Como control. Entonces teníamos nuestros macrófagos cargados. En el meantime de estos PBMCs, teníamos los PBMC, dejábamos una porción de ese mismo donante y las dejábamos 6 o 7 días con antígeno e interleuquina 2 para expandir a las células antígeno específicas. De ahí separábamos las CD8 y las cocultivábamos con los macrófagos. Entonces mirábamos diferentes cosas. Una de ellas fue la transferencia de moléculas citotóxicas desde la CD8 hacia el macrófago. Entonces, lo que hicimos fue tomar el gate de macrófagos, ¿sí? Teníamos, porque es un cocultivo, teníamos los linfocitos de los macrófagos. Marcamos CD8 con CD3. Entonces, en este gate, los doble negativos son los macrófagos que no están asociados a CD8. Éramos como muy cuidadosos para mantener, a nosotros nos interesaban los dobletes. ¿Se dan cuenta, no? Entonces éramos muy cuidadosos para que no se despegaran. Y los que coexpresaban de este gate, vuelvo a decirlo, los que coexpresaban eran los macrófagos que tenían linfocitos T, CD8 asociados, adheridos. ¿Bien? Analizamos granzima y en los asociados vemos que hay y en los no asociados no. Pero hasta ahí nada nuevo, ¿no? Esa granzima que vemos es de los CD8 que están pegados al macrófago. Pero en estos negativos, ¿sí? Estos macrófagos que ya no están pegados al linfocito TCD8, analizamos la granzima en dos condiciones de diferenciación de macrófagos, ¿no? Macrófagos, lo que sería un M0 y M1, y vemos que la estimulación antigénica indujo la aparición de granzima en los macrófagos que no estaban asociados. ¿Está bien? Esto lo corroboramos por microscopía también, que no se las traje. Pero bueno, fue una manera, como nosotros venimos del palo de la citometría desde siempre, fue una manera de utilizar la citometría para contestar una pregunta. ¿Ok? Otra cosa que estamos empezando a hacer es trabajar en metabolismo, entonces obviamente vamos a la citometría y utilizamos un análogo de glucosa fluorescente, el 2-NBDG. El 2-NBDG entra a la célula por transportadores de glucosa. Dentro de la célula se fosforila y queda atrapado y emite fluorescencia en el rango del primer detector, ¿no? De FL1. Con lo cual podemos analizar la incorporación de glucosa de células antes y después de la estimulación. Obviamente esto nos da la posibilidad de hacer infinitas cosas. Porque uno dice, bueno, ¿para qué te vas a hacer tanto problema? Medí glucosa en el sobrenadante, mira cómo baja y ya está. ¿Qué ventaja me da esto con respecto a medir glucosa en el sobrenadante? ¿Qué les parece? Entonces, si puedo ver por célula, ¿qué puedo ver por célula? Puedo armarme un megapanel y ver el fenotipo de cada una de las células que está respondiendo a la estimulación y de qué manera incorpora. ¿Sí? Perdón.
¿Verdad?
A ver. Perfecto. Les decía que se puede hacer un megapanel incluyendo a el 2-NBDG para conocer el fenotipo de estas células que están incorporando glucosa. Son linfocitos T, CD8, son CD4, son linfocitos B, son linfocitos vírgenes de memoria central, de memoria efectora. Bueno, todas esas preguntas las puedo contestar cuando tengo una técnica que me permite ver a nivel de célula individual. El Seahorse es fantástico, pero me dice, me cuenta lo que pasa en el bulk de células. ¿Sí? Ahora lo vamos a ver un poquito. Puedo ver el metabolismo mitocondrial también, como decía Bill, la salud celular. Y acá les traigo un ejemplo nuestro de mitosox. para ver metabolitos oxidativos mitocondriales, cuando desacoplo, este es nuestro control positivo, ¿no? Cuando desacoplo la cadena de electrones, sube el, la marca. De nuevo lo mismo, esto nos permite investigar cuál es el fenotipo en una mezcla de células, ¿sí? Tengo sondas para ver contenido mitocondrial, potencial bueno, todos los que nos contó Bill, que lo podría hacer por microscopía, pero por citometría me parece que es mucho más potente. Y les quería traer también, siguiendo con el estudio del metabolismo, esta técnica que se llama CENI, que la desarrolló un argentino que trabaja en Marsella, que ya varios lo conocemos, Rafael Argüello, y publicó este Cell Metabolism, que es una cosa espectacular, donde muestra que el estudio del metabolismo con esta técnica da la misma información que el Seahorse. ¿Bien? Y es una cosa bastante sencilla si uno lo piensa. Ganó el premio de Cyto el año pasado, creo. El Mary Ingram, por el aporte que hizo con esta técnica. Básicamente uno tiene sus células, vuelta a lo mismo, una mezcla de células. Enfoquémonos en esta figura, ¿no? Tenemos una mezcla de células después de, o puede ser apenas sale del paciente, ex vivo, puede ser después de un cultivo, de un tratamiento, de lo que sea. Se la divide en 4, esa condición, ¿bien? Ese número de células que estuvieron todas bajo la misma, en el mismo well, la divido en 4. Y las expongo a, bueno, una parte la dejo como control, otra parte la pongo en contacto con deoxiglucosa, que es un inhibidor de la glucólisis, otra parte con oligomicina, inhibidor de la fosforilación oxidativa, y otra parte con las 2 drogas. ¿Bien? Lo que va a ocurrir es que va a caer el rendimiento energético de la célula, la producción de ATP. Y la producción de ATP está estrechamente ligada a la síntesis proteica. ¿Bien? Entonces, luego del tratamiento, que el protocolo dice este tiempo, uno agrega puromicina, que es un antibiótico, pero que inhibe la síntesis proteica y se va uniendo a las proteínas a medida que se van sintetizando. Hasta ahí bien, ¿no? Entonces, voy a tener una modulación de la síntesis proteica por estas drogas y un marcaje de la síntesis proteica con puromicina. Una vez que tengo mis celulitas con los tratamientos, hago la marcación. La marcación consiste en utilizar un anticuerpo antipuromicina, que lo desarrollaron en este laboratorio. Este anticuerpo antipuromicina se va a pegar a la puromicina que se pegó a las proteínas que se sintetizaron en este periodo de tiempo. ¿Bien? Entonces, ahora enfoquémonos en este histograma de acá arriba. Y yo voy a analizar la intensidad, voy a graficar histogramas analizando la expresión de puromicina. ¿Bien? Enfoquémonos acá. Las células control van a tener el máximo de expresión de puromicina, ergo van a tener el máximo de síntesis proteica para mi sistema. ¿Bien? Las tratadas con deoxiglucosa, que son las violetitas, van a tener menos síntesis proteica, por ende, menos expresión de puromicina. Y por último, el control de la inhibición total, donde tengo aún menos síntesis proteica. Acá está el caso de la oligomicina, están separados porque se hacen cálculos diferentes, donde el máximo de síntesis proteica está dado por las células control, La oligomicina hace que disminuya la síntesis y lo mismo que acá, la conjunción de las dos drogas da la menor síntesis proteica. A partir de todos esos datos uno puede hacer cálculos. Acá tienen el paper, y les recomiendo que lo vayan a mirar, porque si no voy a estar 3 horas contándole los cálculos, pero uno puede analizar cómo se comporta la célula con la glucosa en cuanto a su dependencia y su capacidad glicolítica, cómo es la oxidación de ácidos grasos y de aminoácidos, la capacidad y cuál es su dependencia de la energía producida por la mitocondria. ¿Está bien? Y puede hacer estos grafiquitos. Pero bueno, yo les estoy contando en un bulk de células. Uno, si incorpora marcadores, como estamos hablando acá todo el tiempo, podemos individualizar las células, las poblaciones de interés, y analizar cuál es el perfil metabólico de esas poblaciones. ¿Bien? Como les decía, hicieron un análisis exhaustivo con Seahorse y bueno, y con otras, midieron ATP, bueno, muchísimas cosas, y vieron que esta técnica paralela eh, es muy fidedigna de lo que ocurre con el Seahorse, que es nuestro gold standard, ¿no? Para mostrarles nada más nuestros resultados donde vemos síntesis proteica total, ¿no? Les estoy mostrando acá. Y siempre, fíjense, siempre en forward, la local forward para ver los cambios morfológicos en nuestras células. Lo que no les dije es que la marcación intracelular se hace, nada, con los kits que nosotros conocemos. Se puede utilizar con kits de marcación intranuclear o con kits de marcación intracelular, intracitoplasmática. No hay ningún problema. Y vemos que las células control tienen poca capacidad de síntesis proteica y aumenta de manera importante cuando son estimuladas, son PBMS estimulados con micobacterium inactivado. ¿Bien? Y estos son unos resultaditos con pacientes con tuberculosis, donde vemos incremento en la síntesis proteica y que una droga que nosotros estamos investigando pareciera que mueve un poquito la síntesis proteica, pero al analizar el perfil metabólico, esta droga nos cambia el perfil metabólico desde aumentando todo lo que es glicolítico, ¿no? La dependencia de glucosa y la capacidad glicolítica. Son preliminares, pero bueno, yo les quería mostrar la utilidad. Y esto lo hicimos en linfocitos TCD4 y lo queremos hacer en diferentes poblaciones de memoria efectora específicas para micobacterias. Recién estamos como empezando. ¿Alguna pregunta? Buenísimo. Todos los PBMCs. Exacto, exacto. Sí, sí. Estos son datos que nos pasó también, que me pasó un colega del instituto, donde investiga la funcionalidad de macrófagos y dentro de la funcionalidad la esferocitosis, que es la capacidad de englobar partículas apoptóticas. ¿Bien? Entonces, y cómo la prostaglandina modula este proceso, ¿no? Ellos quieren, la pregunta es cómo la prostaglandina modula la funcionalidad de macrófagos. Con lo cual, lo que hicieron fue cocultivar macrófagos con cuerpos apoptóticos producidos desde neutrófilos. Y por citometría lo que ven es tomando los macrófagos que en forward y HLA-DR van a ser positivos. De aquí lo que observan es que el tratamiento con prostaglandina de los macrófagos incrementa la capacidad de fagocitar cuerpos apoptóticos por parte de estas células. Esto se los estoy mostrando con cuerpos apoptóticos, pero se puede hacer un setting donde uno use bacterias marcadas con fluorocromo, puede hacerlo con bits, puede, bueno, tenemos diferentes, usen la imaginación, ¿no? Porque pueden hacer lo que quieran. Una partícula marcada con fluorescencia que es incorporada por el macrófago. Algo que tenemos que tener siempre en cuenta en este tipo de ensayos es que el citómetro no nos dice si esta partícula está efectivamente adentro de la célula o está pegada a la célula. Así que ojo, especialmente cuando uno trabaja con bacterias, de alguna manera barrer las bacterias que quedaron en la superficie de la célula. ¿Bien? O corroborarlo por microscopía, ¿no? Pero las técnicas, las técnicas que he visto que trabajan con bacterias hacen eso precisamente, hacen un tratamiento, ya sea con pH o lo que sea, para barrer las células bacterianas que puedan quedar adheridas al macrófago. Bueno, cuando tengan su setting experimental van a tener que pensar en ese tipo de cosas. Y por último, que ya Bill les contó un montón sobre apoptosis, pero yo quería ir, y chicas, ustedes también si me ayudan con conceptos que quizás yo no los tengo tan leídos. La utilización de las ondas para viabilidad. Que Bill les tiró un tip para ver proliferación celular con yoduro de propidio, por ejemplo, uno usa altas concentraciones. Bien, uno lo que quiere es saturar el ADN para que todo el ADN esté pintado igual. Vieron que son picos angostos. Entonces uno satura totalmente a la célula. En este caso no es necesario, porque yo lo que quiero es que la célula se ponga positiva. La célula muerta va a tener poros, el yoduro propio en este caso va a entrar a la célula, va a marcar el ADN y no importa que sea un pico angosto, lo que quiero es ver positivas para excluirlas. Bien, entonces voy a trabajar em, con bajas concentraciones. Pero bueno, eh, vamos con las que son las que nosotros usamos habitualmente, que se unen a grupos amino. No, pará, estoy haciendo algo mal. Las ondas fluorescentes que reaccionan con aminas se unen a proteínas en la superficie de la célula. Este cartoon me gustó porque estas ondas se unen a todas las células, ¿bien? A las vivas se van a unir poco porque van a estar en la superficie. Y a las muertas se van a unir mucho porque las células muertas tienen sus poros y entra y tengo más intensidad de fluorescencia. Entonces, como les dije, se unen a la membrana en las células vivas, en las células muertas en el citoplasma, y podemos ver entonces que las células muertas se van a desprender de las vivas. Con estas sondas podemos fijar y permeabilizar las células, ¿sí? Es una gran ventaja en términos de logística y de adecuación a los protocolos que nosotros utilizamos. Y la contra es que requieren, al momento de la marcación, utilizar buffer libre de proteínas. Entonces quizás se tiene que hacer una marcación en 2 pasos, con lo cual alargaríamos los tiempos de marcación. En cuanto a las que se unen a ADN, que es, por ejemplo, el yoduro de propidio, son yo creo que son las primeras que aparecieron. Son las más económicas, súper prácticas, lo que pasa que son tóxicas. Y lo malo es que no se pueden fijar, con lo cual se recurre más a las anteriores, ¿no? Entonces tenemos el yoduro de propidio, que se une al ADN directamente. Y el 7AD, que también es muy usado, que puede ser, la ventaja que tiene, o sea, el yoduro de propidio ya Bill les dijo que tiene un espectro como muy ancho y se mete por todos lados, es bastante antipático en ese sentido. El 7AD es más simpático en ese sentido y puede utilizarse con FITSI o con PE, ¿bien? Pero la mecánica es siempre la misma, la célula tiene que estar viva, no puedo hacer fijación. Y el DAPI, la desventaja que tiene es que también se une al RNA. La realidad es que nosotros no usamos DAPI, no sé ustedes, me parece que es una antigüedad. Bueno, entonces, em, siguiendo con las diferentes opciones que podemos llegar a tener, el CFCE también se puede utilizar como indicador de la viabilidad celular. En la técnica que yo se usa como indicador de la viabilidad y ya les conté que el CFC es muy lipofílico, entra a la célula, las esterasas quitan el éster y ya queda con capacidad de reaccionar con las proteínas, con lo cual queda atrapado en la célula, con lo cual las células viables las voy a poder ver bien brillantes y cuando se entren a morir o vayan muriendo a lo largo de mi ensayo, voy a poder ver la pérdida de CFC para esto. Y también son, al ser tan brillantes, lo puedo utilizar para traquear las células en animales, ¿sí? Y obviamente para proliferación. Otras opciones son la calceína y el Cell Tracker Deep Red, que tienen el mismo fundamento. Así que bueno, para terminar, ¿en qué pensar cuando vamos a elegir una sonda de viabilidad, ¿no? Si mi muestra la voy a fijar o no la voy a fijar. Porque, por ejemplo, el yoduro de propidio es tóxico, es antipático para el citómetro también, porque puede llegar a impregnar las tubuladuras, pero es muy práctico finalmente, porque uno viene con las células vivas al citómetro, pone el yoduro de propidio, las corre y puede diferenciar muy fácilmente entre vivas y muertas. Y no tengo que estar pasando por un paso adicional de marcado, con pérdida de células y qué sé yo, si estoy utilizando algún otro tipo de sonda de viabilidad. A eso me refiero con considerar los pasos en el procesamiento de muestras. Identificar, obviamente, hacer el análisis como si fuese un anticuerpo, si es compatible con los fluoróforos de mi panel. Tenemos, ustedes ya saben cómo hacerlo y además tenemos herramientas web. Obviamente, saber con qué instrumento vamos a trabajar. Bueno, ustedes ya saben absolutamente todo esto, ¿no? El equipo, las sondas, nuestro panel, considerar el tipo de células que estamos analizando, porque no todas las células se tiñen de la misma manera. Si estoy trabajando con un tejido disgregado, también, ojo con eso. Y a eso viene el tema de titular la sonda en su sistema experimental. No es lo mismo trabajar con células en suspensión, donde las cuento, las veo divinas, pongo la sonda de viabilidad y corre divina, a trabajar con un tejido disgregado, donde esa sonda se la va a chupar todo el debris que tengo, con lo cual sí o sí ahí tengo que titular. Entonces, ser críticos al momento de pensar nuestro experimento. ¿Bien? Así que bueno, con eso espero que les haya servido y muchísimas gracias.
Muchas gracias, Florencia. ¿Preguntas? Fueron surgiendo durante la presentación, pero muy didáctica. Por supuesto que siempre vamos a tener más aplicaciones. Podríamos hacer aplicación versión 4, aplicación versión 5 y 6, porque siempre uno cree que abarca todo, pero siempre hay mucha cosa que queda, bueno, toda la parte de bacterias, la parte de parásitos, parte de proteínas fluorescentes, bueno. ¿Eh? Sí. Sí.
Claro.
O incluso no, porque, por ejemplo, bueno, el análisis de aguas, de algas que tienen su fluorescencia intrínseca, o incluso hay gente que lo usa solamente, como decía Florencia, que muchas veces uno se para en el Forward Science Scatter para decir, bueno, hago la región y me olvido, pero uno es medio insistente porque uno es exitista y va a la fluorescencia, le gusta brillar, pero vayamos a ver la morfología, cómo cambia mi población, cómo, digamos, eso
Es
muy
fundamental.
informativo. Si no
Sí.
hay cambio, bueno, está, no nos preocupemos, pero si hay cambios por activación o por cultivo, incluso sin usar marcador de viabilidad, muchas veces uno, ya mirando el forward side scatter, se da cuenta que pobre, madre mía, mi célula. Entonces, capaz que es eso, es como una especie de entrenamiento. Sí.
Claro.
Exacto.
Los melanocitos a veces puedes incluso sortearlos sin marca, porque con luz ultravioleta tienen melanina y eso te va a permitir distinguirlos. O sea, hay muchas aplicaciones, el ciclo celular, o sea, nosotros hemos tenido proyectos en los que, porque quieren hacer screenings con drogas de metilación y mirar luego estado de metilación, no quieres poner un DNA binding dye, pero a veces simplemente con scatter se puede distinguir las células que son haploides de las células que no son. O sea, no vas a nunca tener una pureza del 95%, pero puedes enriquecerlas. O sea, cualquier realidad el citómetro simplemente trae la muestra, se prueba y siempre hay cosas que se pueden optimizar.
Sí, queriendo interiorizarme un poco en el tema de bacterias y levaduras, tienen autofluorescencias características las diferentes especies. Así que sí.
Nosotros, por ejemplo, hemos hecho también, sin utilizar fluorescencia, sorting de células senescentes porque tiene un mayor size scatter. Se hace sorting y después de eso se sigue trabajando o hacer un sequence o lo que sea, pero sin uso de marcación. Por supuesto, como siempre, hay que poner a punto la condición no senescente, senescente, para estar seguro de lo que uno está haciendo, pero digo, sin utilizar fluorescencia. Preguntas.
Bueno, muchas gracias.
Ah. Antes de irnos a almorzar, me dicen acá por la chicharra que tienen que ir a habilitar las cuentas de FlowShow en las computadoras. Yes. For the practical activity, we will divide in 2 groups. Everything is fluctuating. I will switch them.